Okay, so just real quick, I'm going over the last line of defense, your lymphocytes. So remember, we have those granulocytes that are full of different things like histamine, and then these are the lymphocytes. So these are your like protective and delayed and specific. So specific to whatever pathogen is going on, and it's considered delayed because you kind of have to make the memory for the cells about whatever pathogen it's fighting. So starting with the B lymphocytes, so these grow and mature inside the bone marrow. They're also referred to as the humoral immunity. Whenever they are activated, they differentiate into two different plasma cells. And basically, one is a memory producer that allows for long-term immunity and allows for those antibodies to continue floating around. So kind of what vaccines want and focus on. And then the antibody producers. The antibodies themselves are the things that do the work. So these lymphocytes never even touch the pathogen. They kind of let the antibodies do that. There's 5 different types of antibodies. Each one works in a different way to get white blood cells to either attack it, to aid in white blood cells attacking it, or to actually destroy it itself. 2 ways that I did want to point out specifically, because it is specifically mentioned in the, I believe, the 4th RRD, is neutralization and opsonization. There's like seriously so many different ways that these work, so the fact that they're pointing these 2 out, I did want to go ahead and make sure you know that, because they're probably going to ask that on the test. So for neutralization, the immunoglobulin, which is another word for antibody, binds to the toxin that the bacteria, virus, whatever pathogen has excreted. So if you remember back to micro, sometimes some ways that the, more specifically bacteria, but anything will attack the body is by releasing toxins. So this is one way, this is one of the ways that the antibodies kind of protect the body, is by binding to the toxin so it can't attack the body. For opsonization, it actually coats the pathogen with chemicals that helps your whichever one is eating the pathogen, it like chemically helps to phagocytize the pathogen. So make sure you know those 2, because those were like the 2 that were mentioned. On the other hand, you have T lymphocytes, or your cell-mediated immunity. I'm not gonna lie, I use cell-mediated immunity for both T lymphocytes and B lymphocytes, and I did not know until now that they were 2 different things. So, fun fact about that, T lymphocytes get their name from the thymus. They are produced in bone marrow, but then they migrate to the thymus where they mature. So just fun little fact. There's 2 ways that they deploy, and this is kind of the same with the B lymphocytes. One way is you get those chemical signals from the actual injury site. So anything released by your mast cells and those secondary response like CRPs and stuff like that. Or they can just be on patrol, so to speak. They're just kind of floating through everything, and they recognize the surface proteins. So that's going to be really important for both of them, because that's how they recognize pathogens, is there's different proteins on the actual cell itself, and those are what they remember. And it's kind of like a lock and a key, where if it fits the lock, it activates it, right? So then these differentiate as well into CD4 and helper cells, and then the CD8 and natural killer cells or NK cells. So the CD4 and helper cells are usually the first to arrive, because they are the ones that organize everything, and they do so using cytokines. So they organize the attack from the T lymphocyte. They organize things like antibody response, so your B lymphocytes, and the whole time they're just kind of talking back and forth. So they don't actually do anything with the pathogen, but they're kind of like your, what are they called? Like your airplane towers. Oh my gosh, the FAA and like air control. There we go. Air controls, where they're like kind of telling everyone what to do, and they're doing that using cytokines. There's also multiple subsets to these types of T lymphocytes. Like I mentioned, the antibody response, and it also prevents autoimmunity. So basically it makes sure that the killer cells, the ones that are actually doing the work, aren't actually attacking the body and just attacking the pathogen. That becomes really important when you have autoimmune disorders where that doesn't happen. So then you have your CD8 or your natural killer cells. These are the only cells that actually will engage with the pathogen to destroy it. And I mean that within your B and T lymphocytes. Obviously other white blood cells do that, but that's why they're called natural killer cells and your fighter cells, because they will use different chemicals to kill the pathogen. It also is an innate response to kill cancer cells. If they notice that there's something that has started rapidly producing and those cells are not being controlled, they will kill those too. One thing I do want to point out with your T lymphocytes is that this is what we're trying to suppress whenever we give anti-rejection medications for transplants. So if you remember, I kind of mentioned the surface proteins. So there's a very specific drug that you will see every single patient that has received a transplant of any kind. They will be on this drug called tacrolimus or tacrolimus, or however you prefer to say it, or for short, tacro. It is actually derived from a fungus found in Japan at the base of a mountain. Super dramatic. Anyway, so what this stuff does is basically but it kind of like puts blinders on your T lymphocytes so they can't recognize surface proteins, because anytime they recognize a surface protein that's not part of the body, it's obviously going to freak out and destroy it, because that's what they do. However, transplants, that is not your organ, it is someone else's. The DNA does not match. And so that's where they want to kill that organ. So totally natural, but obviously not great for the organ. So it kind of like hides, it like blinds the surface proteins, which is great, except for like, that's kind of the point of T lymphocytes. So you kind of lose your cell-mediated immunity, which really sucks. The way that we so that's why all of your transplant patients are going to be immunocompromised, or it's easy for them to get sick. The way that we try to best prevent that is by cell typing and tissue typing. So for the person to be a match, they have to be at least 95% similar to the donor in DNA, which is kind of crazy to think, but it's actually pretty common. They call you at the 95% once they hit that, and they basically ask you if you want a transplant. I want to say kidney because that's what my mom had, but if you want to accept the organ, and if you say yes, they say, okay, come to the hospital. And then you spend the next 6 hours hurry-upping and waiting. Yes, I know that was not proper English, but and what they're doing during those 6 hours is that 5%, they're trying to whittle down even more, so we don't have to use as much Tacro and other things that we use to kind of, unfortunately, kill the cell-mediated immunity. So then the closer they are in similarity, obviously, the less likely it is that these lymphocytes are going to want to eat the kidney or organ. So in my mom's case, we were very blessed, and the donor, who we don't know, it was a deceased donor, was actually 98% similar. And so that 2% is made up by those anti-rejection medications. So that's not all on the test. I just kind of wanted to link it back to like why this kind of matters. But yeah, that's your third line of defense, just real quick. And then next I'll probably go over all the things like sepsis and SIRS and what happens when your immune system doesn't work. So see y'all next time.